Okay, testing mic. Test my mic. Okay, so I think my mic is function. Okay, so now we can start our class now. Okay, so welcome back to our class now. So for today, I will complete the chapter 17, Inheritance Genetic Control chapter. So we only left two topics, regulation of gene expression, and another last topic is about mutation. Okay, so without further ado, let us start now. Okay, why? Wait. Berbini. Why? Okay. Okay, so let us start now. Okay, so this is our topic today. So for today we will focus this regulation of gene expression and all the mutation. Okay. Okay, so we go to the regulation of gene expression topic. So actually, we already learned gene expression in yesterday, right? So what is gene expression? Gene expression is to the mechanism our cell will produce the protein, the protein synthesis mechanism. But this is for our cell. Our cell can use our DNA material to produce the genetic to produce a polypeptide change which protein lah. Okay, so actually we have one regulation. Our cells will have the one regulation system to make sure that our gene will be expressed within certain condition. In real situation, our gene will not be expressed all the time one. It will express only in certain situation only. So that is why we say that regulation of gene expression. We need to regulate our gene expression. So meaning dia ada satu control lah. Ada regulation dia, ada control dia. So in our syllabus, we only learn about prokaryotic cells only. Eukaryotic cells we not learn for our syllabus in STPM syllabus. Because prokaryotic is more easy and simple compared to eukaryotic. Eukaryotic is more complex. So of how the prokaryotic cells control the gene expression, it is controlled by one system called operant system. This is the system that exists in the prokaryotic cells. Okay, so what is operant system? So operant system actually is a functional unit that consists of promoter, operator, and structural genes that function as transcribed as a single mRNA. So it is the one of the functional unit. It contains a promoter, operator, and structural genes. Okay, so if you see here, so this is the one of the example of operant system, lab operant system. This one that we learn. In our syllabus, we just learn one system only. So if you know that aware that the definition, it only say that consists of promoter, operator, and structural genes. How about ini promoter right? Ini operator, ini let Z, let Y, let X. Ini semua structural genes. How about this? Ini siapa ni? So R is our regulator. So actually R is not involved in the operant system. It does not involve in the operant structure one. Although it is involved in the operant mechanism, but R does not involve in the operant structure. So please aware this. Regulator is not the lab operant structure. Dia bukan sebahagian daripada lab operant punya. Dia lain dua punya. So actually, this one actually is our DNA, is the DNA strand that contain regulator region, promoter region, operator region, and also structural gene. So for let opera, they contain three structural genes: let Z, let Y, and let A. So these are the enzymes that can be expressed. So let A will express tranacetylase, let Y will express permease, and let Z will express beta galactosidase. So this beta, you need to write this one yang ni punya, bukan macam not this one, ini tak boleh because this one I use I use computer so at the moment I don't know why my computer cannot type this, so I use this lah, this to replace it. But actually in examination you only can write this Latin word, tak boleh guna yang ni. So this is just the lab operant structure, the basic structure you need to memorize, and also memorize the sequence there, especially the structural gene. You need to memorize A, Y, Z. A yang paling kanan, Z yang selepas operator. Awak kena memorize. Karena maybe in structural question dia dah kamu label, and also you need to also memorize each gene express what enzyme. Let Z beta galactosidase, Z let Y permease. So all of these you need to remember it. Okay, so this is all the definition. Okay, so let Z, let Y, let A. I think all these is structure are the structural gene that express enzyme you already know. So operator. So operator is the binding site of repressor protein. Promoter is the binding site of RNA polymerase. And another one, R regulator is to synthesize repressor protein. So what are they? What are their functions? So we go to this mechanism. This mechanism is very important. So let operon. So what it is, we call lab because of lactose. Either this lab operon will switch on or switch off. Dia boleh buka atau tutup je. Based on the presence of the lactose molecule. Either it is present or absent. Okay, so when lactose is absent, tak ada lactose, no lactose at all, meaning that our regulator will express the mRNA, and then will express the repressor enzyme. It will express the repressor enzyme. Okay, repressor enzyme, repressor protein repressor protein. So this protein will directly bind to the operator because it has the same shape with the operator one. It will bind to the operator, directly bind. So if this repressor protein bind to the operator, meaning that the RNA polymerase, so if you still remember, right, RNA polymerase is play the role in transcription process, right? It is the initiation process. So RNA polymerase will bind to the promoter to initiate transcription. But for now, because of operator is already bind by the protein, repressor protein, so our RNA polymerase cannot bind toward the promoter region already. Tak boleh bind lah. The late repressor is innately active, and in absence of lactose, it switch off the operator by binding to the operator. The enzyme for using lactose are not met. So meaning that RNA polymerase cannot bind to the promoter region already. It cannot bind already. So when RNA polymerase cannot bind to the promoter, meaning that no transcription process occur. Right? Tak ada transcription process because RNA polymerase tak boleh bind. So no transcription process occur. So no transcription follow no translation lah, because no mRNA already not produced. No mRNA produced, so no transcription. So equally no translation. So if no transcription, no translation, meaning that the structural genes are let A, let Y, and let Z, all these will not be able to express the enzymes. So no enzyme occur. So this one we call as switch off. itu switch off. The opponent system is already switched off already. No enzyme will be produced by the structural genes. So this happens when the lactose is absent. No lactose in the cells. Okay, so this is very easy. So this explanation you can build yourself. Okay, next one is lactoperon. Alright, when lactose present. If lactose present, how about it? What does it happen? So as we know that our the regulator of regulator will express the repressor protein, right? Will express the repressor protein. So if no lactose present, it will bind to the operator. But however, when lactose is present, got one molecule will be called as allolactose. So allolactose is another isomerization name lah. Isomerize of the lactose. Allolactose have the same shape with the repressor protein. So it act as the inhibitor lah. Normally it act as an inhibitor, bind to the repressor protein to form to make the repressor protein inactivate, deactivate dia. Dia tak aktif lah. So meaning that it already changed the shape of the repressor protein. The shape of the repressor protein is already changed so that it already cannot bind toward the operator. Dia tak boleh bind dekat operator. So meaning that operator is not binding with repressor protein, so operator is totally free at all. So our RNA polymerase will start to can bind the promoter region. So when RNA polymerase bind to the promoter region, then transcription process will be occur. Right, transcription process will be occur. So our LAC-A, LAC-Y, and LAC-Z gene will start to produce mRNA strain. Right, three mRNA strain, different three mRNA strain. After that, translation process will be occur by the tRNA and occur in the ribosome, so that will be produced respectively enzyme. So LAC-A will become trans-acetylase, LAC-Y permease, LAC-Z beta-galactosidase. So these are the three enzymes. What are the function of these three enzymes? These three enzymes is to hydrolyze the lactose mechanism. So want to hydrolyze lactose, these three enzymes is play a responsibility lah to hydrolyze the lactose component. Okay, so this is what we call as our lactoperon is switching off, turn on already. Because no barrier at the operator. So our RNA polymerase can bind to the promoter, so that transcription process will occur, will produce mRNA, and then translation occur, produce the three enzyme. And then these three enzymes will be used for lactose metabolism. So normally, as the important part is here lah, repressor protein. Why repressor protein cannot bind to the operator? Because of the allolactose. We act as the inducer. Allolactose act as the inducer. It induce that our operand to switch on. So if no allolactose present, meaning it like that lah, it switch off because of allolactose. So our repressor protein will bind to the operator, so no RNA polymerase cannot bind to the promoter, so no transcription process occur. For this, because of the allolactose, it acts as inducer, it bind to the repressor protein, cause the conformational change of repressor protein. So the shape of the repressor protein is already changed. So the RNA polymerase can bind to the promoter, and then transcription process occur between these three genes to produce the three different mRNA. Afterward, it will undergo translation to produce enzyme. So this enzyme is used for the metabolism of lactose molecule. So that's all. So this is the explanation. So that's all for this. That's all. So it is I think this one is very easy topic lah. Very easy one. And as long as you know the process of concept of the transcription and translation, if you already know, so this one is not very difficult. So just you need to know that you don't want to terbalik kan lah, reverse that lactose present and lactose absent one. You tambah terbalik kan. Nanti ada cakap, oh lactose present switch off, lactose absent switch on. Terbalik lah, salah lah. So please differentiate between these two lah. Okay. And another one, if you see the Oxford there, it will give another operon called tryptophan operon. But if you refer to MPN syllabus, actually MPN just say you need to learn lab operon only. So tryptophan operon actually you don't need to learn because not in the syllabus. But if you want to know, you can know it. Awak boleh belajar punya. It actually is same je. Dia sama teori je, just the mechanisms tak sama sikit lah. But however, lab operon is the must lah. You must learn lah. Before you learn tryptophan operon, you should learn you should master the lab operon first lah. Okay, so that's all for gene regulation, gene expression regulation. So now next I will proceed to the mutation. Okay, so this is our already last topic lah. Last topic for this chapter. So finally, finally. So mutation. Okay, so mutation topic, I think you already also learned in SPM BIO. You also know that mutation, we have two main types, gene mutation and chromosomal mutation. And gene mutation have four types: substitution, mutation, insertion, deletion, and inversion, right? And then chromosomal mutation, we have two subclass. Either it is change in chromosome structure or change in chromosome number, right? Like structure like inversion, translocation, deletion, and duplication, right? And then chromosomal number, normally you see that the disease all the symptoms one, Down syndrome, Turner syndrome, SO syndrome, Klinefelter syndrome, all this actually you already learned in SPA, just recap lah. Normally it's just recap. So actually this topic also very easy lah compared to the previous topic because you already learned in SPM. So what is the meaning of mutation? So mutation is the abnormal change in the structure or content of genetic material leading to structural and functional change of protein product. So normally mutation is the when there are the abnormal change in the genetic material sequence. Our genetic material sudden change. So if our genetic material, gene sequence, all this is changed, meaning that we cannot produce the functional protein lah, right? Because our gene can produce protein, right? So if our gene is already altered, meaning that our protein it can produce but already cannot function already. So this can make our can make huge effect to human lah. Okay, so now we go to the gene mutation. So gene mutation, what is this? So gene mutation, if already mentioned gene, so it is actually change of the sequence of nucleotide base of DNA to particular gene. So meaning that gene mutation is only occur in the gene. In the gene that is the nucleotide base. All of the nucleotide base ATCG, ATCG or AUCG. Normally it's ATCG lah. Our ATCG to nitrogenous base is already sudden change. So there are four types of mutations. First one is substitutional mutation. So substitute. Okay, substitute. So substitute meaning replace. Dia sudah buat replacement. So if you see here, this one is example. So you need to know the example. Every type of mutation, you need to know the example. So one of the example is sickle cell anemia. So meaning that if you see here, actually normal DNA, normal DNA is CCT GAG. Okay. However, but if you see here, you will found that CCT will become the T here already become A. A here already become T. Actually it should be A. Actually it should be A, right? Sepatutnya kena A, tapi dia sudah tukar kepada T. So if you change this, So if you change this, so the mRNA also will change. So if you actually original one is CUC, but for now mRNA already become CAC already. So if you see that amino acid sequence for respectively, so I think this one is typo. Actually, these two should be same. So actually, it's glutamic acid lah. Both are glutamic acid. So this one also glutamic acid. Yeah. Okay. So actually, it will be expressed CUC to express glutamic acid. However, but if you do the substitution, so this one already changed. So it will change to valine. Amino acid already changed already. It already changed already. So you don't want to see that only one change only. Hanya satu je, tapi it already give the big effect to human already. So it will cause sickle cell anemia disease. So what is this? Sickle cell anemia, I think all of you know. It means that our red blood cell is in the sickle cell shape, right? This kind of people, our red blood cell is contain, yeah, dia darah merah macam bulan punya. The shape is like the moon one. So this kind of people cannot do the exercise one. Dia tak boleh buat jogging activities senaman punya kerana dia tak boleh carry the red blood cell of this patient cannot carry more oxygen. Because the surface area of the red blood cell is very low, so hemoglobin is also very low. Okay, so this one is the substitution. Another one is injection. Okay, so injection meaning that dia tambah satu lagi. Dia tiba-tiba tambah satu nucleotide. Tiba-tiba tambah satu nucleotide base. So you see here, normal one is AUG, AAG, UUU. However, you see here, this G, dia tiba-tiba tambah C dekat sini. Lepas tu G akan pergi ke tempat lain. Kan? Dia akan pergi ke tempat lain. So this one actually is very serious. So if you do like that, if it occur this kind of mutation, it will directly have the many effect afterward. So this one we also can say as the effect is friendship mutation, friendship one, because it already give the huge effect. What is the friendship mutation? Later I'll explain. But this one will give the effect of friendship mutation. It will give the very huge effect. Why? Because you insert C at there, not only this amino acid change, others forward one, afterward amino acid all are changed already, totally changed already. So you see here, actually should be lysine and phenylalanine, right? But for now, it already changed to asparagine and also valine already. Not only here, afterward, all are changed already. All together change. So why all together change? So if you know that genetic code cannot be overlapped. Dia tak boleh overlap. So if you insert, meaning that this is the new genetic code already. So G will be go to another genetic code. So the genetic code all the genetic code sequence are changed. So our amino acid also change. So our amino acid change, our protein also will change. So injection mutation is very serious, I can say, compared to this one. Because this one only can change one amino acid. But for this one, it will change not only one, but afterward all of it. Afterward mutation will change all the amino acid. So this one is very serious. This one also very serious. Deletion. Deletion meaning delete. Salah satu sudah delete. Meaning that this original nucleotide base pair has been deleted. So if you see here, actually AUG, if you see here, AAGUUU, but this A, tengah A ni, dia sudah delete lah. Telah delete. So if it is already delete, so our amino acid become AUG, AGU. So this U is go to backward lah, to form another genetic code yang baru. So AUU, and then this one will form another genetic code. So meaning that this one also will change entire genetic code after mutation occur. Semua amino acid akan berlaku perubahan, effect. So meaning that all the amino acid will change also. So lysine it already change to serine, and then phenylalanine dia sudah tukar kepada yang lain based on the afterward sequence lah. So if you see here, deletion and insertion are very serious, and it will give fan shift mutation effect. Memang akan bagi effect macam tu. So it will give the very huge impact lah. And another, this one is last one, inversion mutation. So inversion meaning that it is the nucleotide base is inverted to each other. So if you see that, sepatutnya ACG-CUA. But if you see here, it changed to AGC-GUA. This C and G is inverted already. Dia sudah buat reverse. Sepatutnya G first, tapi sekarang dia sudah tukar kepada C. Then ini G dia balik kepada belakang. So this one will change the two amino acids. So asparagine and also LEU is leucine, but already forget LEU is leucine. Already forget LEU is what, but however you see that these two amino acids already change. So this one it will not give it, need to see what are the how many inversion occur. So if only one inversion occur, normally it's just only two amino acid will change lah. But however, if there are many inversion occur, so it will also give the very huge effect lah. But this effect does not large like this. But also can give the impact also to the human, because got mutation meaning dia akan ada impact. Okay. So now that this for now you will go to effect of mutation. So it is the effect. Tadi yang ini empat ni, it is for is the type of mutation. Type of mutation. Yang ni adalah kesan, effect dia. Apa effect dia? Okay, so yang ni lah saya akan sentiasa cakap dia. Fan shift mutation. So what does this mean? Meaning that when the number of nucleotides are added or deleted, they are not in number of 3. Meaning that you will see that our deletion and insertion, it can be either insert one nucleotide or delete one or insert two nucleotide. Dia bukan buat tiga punya. Injection not only one nucleotide, maybe it can one, it may two. But if it only involve in one or two nucleotide, actually it will give very huge effect. Because I already say that if you only insert 1 to 2 nucleotide, it will affect entire change of the genetic code sequence. But if you add directly 3 nucleotide into here, so this one will not give the very big effect of the amino acid sequence. Why? Because if you directly give 3, meaning that it just tambah satu lagi amino acid. Just add one more amino acid. So maybe it will not give some huge effect like this. So this is the definition of fan shift mutation. So normally fan shift mutation will be occur, normally is injection and deletion lah. Injection deletion memang akan berlaku. Tapi inversion and substitution tak berlaku. Because inversion and substitution, the change is not very huge. Okay, and then what are the effects? So if you see that these are the effects, provide extreme harmful effect to the protein structure. So the protein structure totally change. Normally it will denature one. The protein already cannot function properly. And then all the amino acid sequence change, and the protein will denature, which totally is unfunctional to the cells. Okay, next one is silent mutation. Silent mutation is the effect that if the mutation occur that does not change any DNA sequence. So this one only can be occur only substitution and inversion only. So meaning that if you go to substitution, meaning that if let's say the substitution occur at here or at here, although it already got mutation, but if you go to the amino acid sequence, actually it will also express the same amino acid. Sama je. If you know that one amino acid can be coded by different type of genetic code, right? So if the mutation occur and the final amino acid expressed actually is same as the normal one. So this one is we call as silent mutation. Tak ada apa-apa. No, nothing else. No effect. Does not give any harmful to human. Because this mutation, although it is occur mutation, but this mutation does not give any effect at all. Amino acid still produce the same amino acid, and then the protein also was remain same. So it is just protein also can function normally. So this is what we call as silent mutation. Okay, another one is missense mutation. Okay, missense mutation is that change in the base sequence alter a codon so that the one amino acid in the protein is replaced by a different amino acid. So meaning that this one is that when the base sequence is changed and then only one amino acid is replaced by different amino acid. So this one yang ni lah, memang. Yang ni memang satu example lah. Kerana sepatutnya glutamic acid. Lepas tu, after substitution mutation, dia sudah tukar kepada valine. So, this one what we call as missense mutation. Another one, nonsense mutation. Nonsense is that when the sequence change to the stop codon, either these three, UAG, UAA, or UGA, directly. Yeah? Tukar, lepas tu directly go to the stop codon. So maybe actually this protein, this mRNA in normal situation can produce maybe 1000 amino acid. But because of mutation occur, nonsense mutation, effect dia adalah got the stop codon. It may be the mutation occur, suddenly at the tengah-tengah part, dia sudah tukar kepada stop codon. Dia ada stop codon dekat sana. Maybe at one mRNA, they have the 10,000 sequence and can produce 1,000 amino acid. Maybe lah, example, 10,000 sequence can be produced maybe 3,000 amino acid. 3,000 amino acid. And then suddenly at the number 5,000, it already changed to stop codon. So meaning that that amino acid sequence cannot produce 3,000 lah. Maybe until 400, 500, that should have stopped lah. Meaning that so that this amino acid cannot change, cannot form the protein shape lah, because the amino acid chain is too short, terlalu pendek. So dia tak complete. Actually the translation process is not completed. So normally this one is substitution lah. Substitution can occur. So this one can provide very serious effect. Compared to these two, this one will produce very serious effect. Because this one, although already changed, but normally the protein still maybe can function. Maybe. But this one is totally no protein form at all. Because the component is not completed. Totally no protein form at all. So protein cannot function because no protein. Okay. And then missense ni dia akan provide some lah. The harmful effect also have, but some tak serious sangat lah. So it maybe can change the shape and structural function protein lah. But compared to these three, it will not give very big effect lah. Compare these two. Silent mutation does not give any effect. Just normal one. Dia macam normal punya. Okay. So this is the effect of mutation. You need to know this one because MPM dia akan keluar ni punya. Dia tak akan keluar ni punya. Ini awak dah belajar dekat SPM, dia tak akan keluar. Dia akan keluar ni. Okay. So that's all for gene mutation. So any question? No question. Okay, so now we will proceed to the last topic, chromosomal mutation. So this is our last topic for this chapter. So finally lah. Okay. So chromosomal mutation is more complex compared to gene mutation. Chromosomal mutation we have two classes again. Under chromosomal mutation, you also have two classes. Either it is change in chromosome structure or chromosome number. right? Like chromosome structure is, we know that our chromosome is look like this, right? Kenapa dia macam ni ah? Baik ni ah. Ayo, I need to. Okay. Okay. You see that our chromosome is like this is our chromosome, right? Ini chromosome kami kan. So if you see that chromosomal structure, meaning that it may be changed at here. Karena our chromosome contain many gene, right? Contain many gene, right? Yang ni maybe dia akan di sini adalah A, di sini adalah B, di sini adalah C, and then di sini adalah D, di sini adalah E. Maybe change in chromosome structure, meaning that this A, B, C, D, E akan buat effect. It will affect. So either A to B ke, C to B ke, atau dia terbalik ke, macam tu lah, macam-macam lah. Later you will learn lah. Okay, this one is about chromosome structure, and then this one is about chromosome number. So I use the example in human lah. So our human contains 46 chromosome, right? Maybe if got mutation, maybe some human only have 45. Some human got 47, right? Either 45 or 47. Some human got extra one. Some human maybe kurang satu. So this one we call as chromosome number mutation lah. So okay, so this is just the generation. Okay, so what are the chromosome mutations? So easy that either it change of number or change of structure. And then so change in chromosome structure, so meaning that this is about chromosomal abbreviation. So meaning this will produce the variety of genetic disorder lah. Okay, so compared to the gene mutation, if the mutation is occur in chromosome, it will give more effect lah, large effect compared to gene mutation. Because you need to know that our chromosome, one locus, ini we call as locus, right? One locus actually contain many genes. Ada banyak genes yang banyak punya. Not contain many genes, contain entire genes. So this one is actually gene A, gene B, gene C. Entire genes is totally changed. Tadi gene hanya change in nucleotide je. Hanya tukar nucleotide base je. Yang ni sebahagian gene sudah tukar lah. Sebahagian sudah tukar lah. Tukar lah. So ini beza dia lah. Okay, so first start is about inversion. So inversion ni dia macam it's same as the gene mutation. So inversion meaning that our gene is totally changed tempat. Dia terbalik. So you see here like this, A, B, C, D. And if you see here B, C, D ni, dia sudah tukar tempat lah. Terbalik, dia sudah terbalik. Make the B ke bawah. And then C still dekat middle, but D ke atas lah. Become A, D, C, B already. So the B and D is already changed location. It already make a reversion lah, terbalik lah. Dia sudah terbalik. And then there are two types of inversion lah. Either it includes centromer or not include centromer. So centromer is that the central part of the chromosome, right? So either it include or not include lah. So pericentric is not include, pericentric is include. Okay. Another one is translocation. Okay, so translocation, what are the beza? What are the difference between inversion? Dia macam sama je. So translocation is that the segment of chromosome is detached and then reattached to different position or same chromosome or other chromosome. So compared to inversion is it detached and then reattached at the same location. Tempat yang sama. Maksudnya awak tengok macam ni, B, C, D dia tukar, dia sudah kedua, lepas tu dia buat terbalik je. Dia terbalik tapi masuk ke dalam lokasi yang sama. Dia bukan ke lokasi yang berbeza punya. Dia bukan B, C, D ni, dia sada-sada tukar kepada sini. Yang tukar lokasi adalah translocation. Translocation, it will change the location. Right? So there are three types of translocation you need to know. Non-reciprocal and reciprocal. So non-reciprocal we also have non-reciprocal intra or non-reciprocal inter. Okay, what are the breakdown here? Intra meaning that intra it only involve one chromosome. Inter it will involve what we say as homologous chromosome, a pair chromosome one, or not only a pair, two chromosome one. Reciprocal also involved in two chromosome one. Okay, what are the beta there? Non-reciprocal intra, it only involve one. So meaning that the gene B, you see here, gene B and C, dia keluar, lepas tu dia insert balik same chromosome, but insert ke tempat lain lah. Insert dekat at the belakang of the F. At the belakang F. So ini intra. Easy kan? Another one is non-reciprocal inter. So inter apa tu? Inter is that it will change to the another type of chromosome. Okay, it will change to the another type of chromosome. So meaning that this BC is keluar, detached, and then reattach to another type of chromosome. So if this case, it will go to the another type of chromosome after N. So BC dia sudah dekat sini lah. So BC like dekat kiri punya chromosome dia tukar kepada kanan punya, dia insert to the kanan punya chromosome, the right hand side chromosome. Okay, and then this one is non-reciprocal. Okay, how about reciprocal? Reciprocal meaning that it is do the exchange of both chromosomes. So if you see this one, B1 non-reciprocal, it only involve the mutation only involve the change attachment. Sorry, the attachment and reattachment only involve one chromosome only. This right hand side dia tak ada buat apa-apa. Dia tak ada detach and reattach, tak ada. Hanya ini kiri punya buat detach and reattach. But for this one, both buat. Meaning A, B, dia tukar kepada yang kanan punya, and at the same time, M and O, dia pun detach and then reattach to the kiri punya chromosome. Dia both lah, dia macam exchange material lah. Exchange the gene sequence. Yang ni dia tak exchange, dia hanya bagi. Contoh lah, yang ni B punya non-reciprocal, dia bagi je. Saya bagi duit kepada awak. Yang ni reciprocal dia tak bagi, dia buat macam pinjaman lah. Saya pinjam awak, then you bawa balik kepada saya. Dia buat macam pinjaman lah. Exchange one, dia buat exchange material. And then this one does not involve exchange. So this is the differences between these three. So please remember it: non-reciprocal, intra, reciprocal inter, and reciprocal inter. Okay. So this is about translocation. So please don't confuse it, and also don't confuse with the inversion. Dua-dua dia macam sama, tapi actually totally not same. If you know the definition, you will know that it is totally different. Okay, another one, deletion. So this one I think very easy lah, delete. Dia delete same as gene mutation. So one of the segment, one of the gene totally delete. Is deleted lah. Dia sudah lost lah. Okay. Another one duplication. So duplication, what is mean by duplicate? Meaning copy lah. Copy satu yang sama, totally sama. Copy and paste. Duplicate is copy and paste lah. So it is that say that chromosome contain more than one copy of particular chromosome segment in homologous chromosome. So if you see here, actually A, B, C, D, E, and then B and C, dia buat copy. B and C copy dekat sini. So meaning that after mutation, it will contain A, B, C, B, C. A, B, C, B, C. So B, C already made a copy lah. So this is about the duplication. So hopefully you guys know. So normally this one, translocation, maybe can make you confused lah. But actually it's very easy. I hope that turns out I explained it well. So if you don't understand, just type in the chat box. Okay. Next one is about chromosome number. So got question? No question. Very good. Okay. So about the chromosome number. Okay, chromosome number yang itu semua Turner syndrome, Down syndrome tu lah. Awak pernah belajar juga. Okay, so but in chromosome number in STPM you will learn about aneuploidy and also polyploidy. So what are they? I will explain it. Aneuploidy is about condition where zygote have an abnormal number of chromosome during fertilization. Fertilization between aberrant gamete with normal. So aneuploidy normally it involves the abnormal number of chromosome during fertilization. During the fertilization, meiosis process lah. During the meiosis process already have the abnormal meiosis process already. So if you see here, so this is what we call as non-disjunction. Non-disjunction in meiosis 1 or non-disjunction either in meiosis 2. So I hope that everyone still know about the process in meiosis 1 and meiosis 2, although in SPM not learn, but this one is you need to know the concept from the meiosis process lah. So normally in meiosis process, so normally the non-disjunction occur. So for this one, what does this occur? Ini phase apa? Still ingat tak? Meiosis 1, meiosis, mitosis, meiosis, we have 4 phase, right? Still ingat tak? Tak ingat, please refer to SPM. Go to SPM, not STPM lah. Go to SPM. Go to SPM. Or go to matriculation. Matriculation ada belajar. STPM tak belajar. So, please know what are these phase. So, you see that, refer that, meiosis 1 and meiosis 2. So, if the non-disjunction is occur in meiosis 1, so you see that actually our chromosomes should be separated. Should be separated to the opposite location already at this phase. So what are these phase? You belajar sendiri. Saya tak mahu cakap lah. Kerana ini sepatutnya awak pernah belajar. So you should know lah. Okay. Okay. For this one, so if you see that this chromosome, these two chromosomes, they do not separate. They are not separate. They are directly attached to each other. So this one will give the result does not give this result. It will this kind of result be in meiosis 2. So if you know that in meiosis 1, normally if you will get 2 cells, right, and you will get the 2 cells with the chromosome of this one and this one. Maksudnya dia akan macam ni lah. In meiosis 1, the product you should get the yang ni satu and then ini satu. Lepas tu ini satu, ini satu. Sepatutnya. Tapi sekarang dia sudah tukar kepada ini satu yang lepas tu ini dah. Lepas tu salah satu hanya satu je. Should be supposed that after meiosis 1 the product of the daughter cell should have both have 2 chromosomes. Sepatutnya. But for this one it has 3 chromosomes and 1 chromosome. Meaning that already. So this one we call as N. This one we call as N plus 1 lah. And then this one is only N. N minus 1. Sepatutnya. Okay, so okay, after that, this is meiosis 1, and then when we undergo meiosis 2, you will found that this one got 3 chromosomes. Right. Karena dia lepas tu chromosome akan buat separation again, right, in meiosis 2, and then this one separate. So finally you will get this kind of result. So all the gamete that will occur in meiosis 1, when meiosis 1 is non-disjunction is occur in meiosis 1, all the gamete contain the chromosome number is already abnormal, totally abnormal one. Karena awak kena tahu kau, ini because in examination maybe MPM dia akan keluar macam ni punya, especially objective question. Dia akan keluar statement, maybe banyak statement lah. Banyak statement, lepas tu maybe based on that statement, we want to ask you, in meiosis 1, is that only contain normal chromosome or not? Normal gamete or not? I don't know. And maybe in structural question, dia nak kamu drawing or want you to label this. After non-disjunction meiosis 1, what will happen? N plus 1 ke? what are the chromosome number change? Or n plus 1 ke, n minus 1 ke, or normal 1, or n minus 2, n plus 2, something like that lah. Itulah, itu semua awak kena tahu punya. Memang kena tahu punya. So that is why that I don't know STP MPM tu lah. Dia buat syllabus and then you don't learn about meiosis. This one you need to know the concept of meiosis so that you will know that why that in meiosis when the non-destruction occur in meiosis 1 n plus you will get this kind of chromosome. So normally all the chromosome, all the gamete will not have the normal chromosome. It either are n plus 1 or n minus 1. Maksudnya salah satu in human, salah satu adalah 24 ataupun 22. Yang tak ada 23 punya. Yang normal one is 23, right? N is 23, normal one. But for when non-destruction occur in meiosis 1, either N equal to 24 ataupun 22. Dia tak ada 23 normal punya. Okay, ini meiosis 1. After that, in meiosis 2, it will give this result. This is the normal result that we want. Ini normal result yang inilah normal result yang kami nak in meiosis 1. But however, when it occur in meiosis 2, it already have the non-destruction lah. Mutation already occur. So mutation occur, so maybe it will occur at here. Nanti. Sepatutnya dia akan separate. All of the chromosome will be separate. But for now, this chromosome does not separate. This one, that does not separate. So they remain. So because only one is non-separate, yang ni still normal, yang ni dua still normal, so it will form the normal. So that is why that you see that there are two gametes are normal one. And then after that, when it is completed, it will produce these two gametes. So one of the gametes is only have one chromosome, so n minus one type of one chromosome, n minus 1, and another one will have extra one chromosome. So this one for meiosis 2, it will have 23, 22, 23, and 24. All of them have their probability. But there are 50%. 50% is normal, and then 25% you will get at normal one. So half lah. 50% awak akan dapat normal punya and 50% at normal. So this is the non-destruction in meiosis 2. But in meiosis 1, totally no normal at all. No normal one. Meiosis 2 ada normal lagi. Okay, so this is the concept you need to know about the non-destruction occur in meiosis 1 and meiosis 2. So this one, please, if you already forget at all what are the meiosis, please recap SPM syllabus. Tolong. Memang lah. Karena saya sini tak belajar SPM lagi lah. Belajar SPM lagi lah. You should already score SPM very well already. Okay, so this is just how the aneuploidy happen. Ini adalah aneuploidy happen punya proses. But however, so there are many types of aneuploidy. So normally in human we have monosomy and trisomy. Normally in human we only have monosomy and trisomy only. So monosomy is that our chromosome we less one chromosome. Trisomy meaning we have extra one chromosome. And then there are also tetrasomy and nullisomy. So tetrasomy is extra two chromosome. Nullisomy is that minor less two chromosome. So for this one will not occur in human. It will occur in some animals lah. Different type of animal. Normally in animal. Human tak akan macam ni punya. Okay. So if human occur like this, normally this human cannot survive lah. If you know that if you even though human is already have less one chromosome or extra one chromosome, it already have huge effect already in our health. So if kalau tambah lagi dua ataupun kurang lagi dua, memang human tu tak boleh survive lah. Memang lah. memang lah. So this one is also maybe occur in the non-detection in meiosis 1 also. So maybe it will occur that something like macam ni lah. Not only one part of them, maybe both of them got non-detection. And then maybe some of them, maybe meiosis 1 and meiosis 2 got non-detection combined. Bukan meiosis 1 je. Meiosis 1 and meiosis 2, both of them have non-detection. It may be occur one, it may be can occur, but you don't need to, in our syllabus, you don't need to learn about all of these. But you want to learn, actually it's very easy. When you know about the meiosis concept, then meiosis concept, actually you will know about all of that lah. Apa-apa sahaja lah. Either meiosis 1, meiosis 2 atau tambah. And then maybe some of them, all of them got non-detection. Dan semua awak boleh main lah. So boleh main lah. So yang ni paling penting awak kena tahu meiosis konsep lah. So kepada siapa yang tak tahu lagi, please recap your SPM lah. Yang lepas tu kalau kamu tak mahu recap, hafal je lah. Saya tak boleh buat apa-apa lah. Memang tak boleh buat apa-apa lah kerana saya nak bagi awak. Kerana awak yang belajar, bukan saya yang belajar. So I already give you a clue already. So when you want to either nak buat revision dekat SPM or not, up to you lah. Okay, so this one tak on including. Okay, so this one is about the non-detection that will occur in oogenesis. What about when the non-detection is occur at oogenesis? What happened? Okay, so if let's say non-detection is occur at female, tak kisahlah meiosis 1 or meiosis 2, it will produce the egg with like this. Either SS, SS meaning that this one will produce M plus 1. Yang ni langsung tak ada. No chromosome at all. No chromosome. No chromosome at all. This one is about the sex chromosome. What we say as sex chromosome only. Why? Because meiosis process only involve the gamete only, right? Please remember. Right. In our autosome punya chromosome, autosomal image, you will know that our human got 46 chromosome, right? 46 chromosome and 44 plus XY for male and 44 plus XX for female, right? Female, right? And then the non-detection only occur in this chromosome only. So these two chromosome only. So either it contain M plus 1 punya, and minus 1 punya, maksudnya dia ada extra 1 lah. Extra 1 punya. So meaning that it will have 23 plus XX. Sepatutnya normal punya adalah 23 plus X. Ini normal punya. After meiosis process, you will get this. Sepatutnya. Tapi in non-normal, dia akan jadi macam ni. 23 plus SS. Another possibility is totally does not have sex chromosome at all. Only have 23 chromosome only. 23, that is autosomal chromosome lah. Sex chromosome totally not have. So if you do this, after that you go to fertilization. So if the female have the non-detection, female is and male is normal. So if male normal, it will produce a sperm either have X chromosome or either have Y chromosome. So afterward, we go to fertilization, you will get this kind of child. Normally all are abnormal. Memang abnormal punya. Semua abnormal punya. Tak ada yang normal punya. Sangat kesian lah. Kalau female ada non-detection. Google non-detection. It will produce triple X female. It will produce XO female. X0 female. Only have one X chromosome. It will produce the boy that have XXY punya. And then also have XY0. So Y0 is the non-viable lah. Dia tak boleh survive punya. Memang during the zygote or during the embryo state, it already have the problem already. So this one cannot survive. This three can survive, but the lifespan it also cannot very high lah, very long lah. Later we will learn this all syndrome. So this one is the possibility that the child will be formed after when the female have the non-disjunction. When for male is bapa dah biasa tapi ibu tak biasa. Okay. So yang ni anak semua bukan anak yang biasa lah. Semua anak ada disease punya lah. Ada genetic disease punya. So sangat kesian lah. Okay. So for this one is about female. Okay. Female punya, kalau for female punya, you will see that when non-disjunction is occur, so for female is more complex sikit compared to female. Why? Kenapa? Oh, unit female punya hanya dua lah. Dan female punya kena separate into meiosis 1 and meiosis 2. Kenapa? Siapa tahu? Oh, genesis. Tak tahu ke? Okay, I don't want to give answer. You forget again process oogenesis. Semester 2. Or not semester 2 lah, SPM lah. SPM pun dah belajar kan. Semester 2 and SPM. So for those who retake U2 punya, boleh fikir and then apply it. Kenapa female kami tak separate into meiosis 1 and meiosis 2? Kami tak separate. Kenapa? But male kami separate. So, I give you answer. If you question, you find it yourself. Okay. So, for male, when the spermatogenesis is not occur non-disjunction, either is meiosis 1 or meiosis 2. So, if let's say non-disjunction is occur in meiosis 1, it will give you this chromosome. Maksudnya salah satu contain XX YY. Sepatutnya hanya XY je. Sepatutnya hanya XY, tapi sekarang dia ada XXYY. XXYY. So I don't know. I don't know. And then after that another one is does not have any, tak ada langsung lah. Memang tak ada langsung. After that, if you go to meiosis 2 is normal, it will give you like this lah. XY. Sepatutnya sperm or either it only have X or Y. And then after meiosis 1, it also either have 2X or 2Y only. Sepatutnya in normal either have 2X or 2Y only. Sepatutnya. But in this non-disjunction occur in meiosis 1, dia akan semua sudah dekat sini. Yang ni langsung tak ada sex chromosome. 0 at all. So in meiosis 2 also this one will also get 0 lah. So this sperm does not have the sex chromosome at all. And then this one have lah. And then so this sperm will produce XY punya chromosome. So itulah. Okay, so when non-disjunction is occur in meiosis 2, dia akan produce like this lah. Dia akan produce XXYY. Memang pun tak normal lah, XXYY. So and then another 2 does not contain any sex chromosome lah. Either dia ada sex chromosome either XXX or YY. So after that, when you go to fertilize with the normal female, so our normal female is XY. So it will give you XY will go to if you XY tambah X, it will give you XXY kind factor male. And another one, if you go to XX, go to XXX triple X, maybe, and then another one is XYY. It will go to XYY. So also cannot form the normal punya lah. So in conclusion, if I not ever your bapa ke atau ibu ke atau have non-disjunction occur, so memanglah itu anak-anak mereka tak akan ada normal punya. Memang ada disease punya. Okay. So now we go to the genetic disease. Yang paling famous, Down syndrome. Memang famous lah Down syndrome. So Down syndrome actually is not involve sex chromosome, it involves the 21st chromosome. In the 21st chromosome, it contains 3 chromosomes. So 21st, it have the 3 chromosome punya. Sepatutnya kami hanya ada 2 je, 2 chromosome, dua homologous chromosome je, tapi yang ni dia ada 3 chromosome. So what are the symptoms? So you see that normally short stature, orang ini memang pendek punya, fall in eyelids, mata dia akan macam mana cakap lah, tak tahu lah macam mana cakap, you see the gambar lah, memang mata dia tak sama, and short nose and short neck, protruding tongue, spot in iris, low resistance to infection. So normally the immunity of this kind of people is very low lah, so they are easily to be infected by outside punya pathogen lah, sangat very easy to get flu lah, the mum. And mentally retardation, so normally this kind of people, the IQ, we say IQ lah, dia tak bijak macam normal punya people lah. So this kind of people, they want to learn something, it takes many time, more longer time lah. Dia nak fahamkan sesuatu benda kena take time lah, bukan macam kami boleh fikir dengan cepat. And then high risk in leukemia and Alzheimer's disease and short lifespan. So normally this kind of people, but how nowadays already have high technology medicine lah, already have medicine. So normally this one people, I don't forget, I think already can survive until 50 years old, until 60 years old pun okay lah. Tapi but that one, unless you get the medical treatment lah, awak ada buat rawatan. Tapi kalau awak tak buat rawatan langsung, memang orang ni memang short lifespan. Okay. So this one is characteristic. So this one you need to know, because in MPN dia maybe akan keluar, dia keluar macam give you some characteristic, and then based on that characteristic you need to know what is this syndrome, what kind of disease it is. So you need to remember. Okay, another one is Patau syndrome. Okay, Patau syndrome is another disease that involve autosome chromosome. It involve at the 13, nombor 13 punya chromosome. Nombor 13. At the 13 punya. So yang ni 13 punya chromosome dia ada same lah, 3 chromosome. So sebetulnya normal punya ada 2, tapi dia ada 3 chromosome. So you see this kind of people, yang paling penting inilah clade leaf, we known as clade leaf or palate. It can have head leaf. Dia macam dia ada lubang dekat. Dia macam hidung dia connect with the mouth punya. Sepatutnya hidung dan mulut kami adalah biasa kan, bukan tak akan ada lubang macam ni, tapi dia ada lah. And then small head, absence of eyebrow, dia tak ada eyebrow punya. And then mental imbalance lah. Normally this kind of people very kesian lah, mental imbalance. Hearing deficiency, tak boleh dengar baik-baik punya, dia memang even tak boleh dengar pun. Some of them maybe can dengar, tapi dia macam orang tua lah, awak kena survey baru boleh dengar. Tapi some of them maybe memang dia original tak boleh dengar langsung lah. Why? Because of the ear. The ear is already not abnormal. Cleft lip, heart abnormality, yang ni jantung dia pun kurang, jantung dia pun sangat berbeza lah compared to the normal people. So because of the heart abnormality, so normally this one very short lifespan lah, 3 bulan atau 1 tahun lah, memang akan mati lah, kerana jantung yang jantung paling penting lah, kerana jantung dia sudah tak kuat sampai macam normal people. So very kesian lah, orang ni memang sangat kesian. And also the testis for male lah, if this patient baby is male, the testis is also abnormal. So very kesian lah, sangat kesian punya. So okay, this is Patau syndrome. Another one is Edward syndrome. Okay, Edward syndrome is occur at the 18 kromosom nombor 18. Yang tadi nombor 13, sekarang nombor 18, so also have 3 chromosome. Okay, so what are the characteristics? So the most characteristic is the small nose and mouth. Small nose and mouth. Hidung dia dan juga mulut dia sangat kecil punya, and then short neck. Dia neck sangat kecil, pendek je. Also mental deficiency, and then the kidney is U-shaped kidney. Our normal kidney is bean shape, right? Bean shape. But it is U-shape punya kidney, and dia deformities, yang ni memang tak boleh dengar langsung lah, sangat kesian punya. And then many organ incomplete at birth, meaning that this kind of baby actually their internal organ normally are not major at all. The organ is incomplete form. Maybe not function lah, so meaning that the function of the organ of this baby cannot function well. And also you see that clenched hand, and then fixed big toe, and inilah semua ni lah. Sangat kesian punya. And then this one normally also 1 tahun lah. Hanya 1 tahun punya. So memang kesian lah. Okay, then now we begin to sex punya chromosome. Tadi semua yang ni semua punya autosomal punya. So actually banyak lagi lah, but ni just in syllabus lah, awak kena tahu punya. Actually if you go to further, so some of them want to go to genetic punya disease, want to further study genetic disease, then you will learn more lah. Not only this, also for now this one with involves sex chromosome. So one of these is Klinefelter syndrome. So this one is about the sex chromosome is XXY. So memang tak tahu lah, memang tak normal lah. So because in normal either it is XX or XY only, either dia girl ataupun boy. Tapi yang ni dia nak cakap dia siapa? Boy ke girl ke? Ini siapa? Boy ke or girl ke? So inilah abnormal lah. So if you see that it is a boy, dia ada testis, but the testis is very small. And then that testis cannot form sperm punya. So no spermatogenesis. So it is infertility lah. Dia tak boleh buat seks punya kerana dia tak boleh form the sperm. It cannot form the sperm punya. So yang ni and then unusually tu yang ni memang sangat tinggi lah. Dia orang ni lelaki ni memang sangat tinggi punya. So for those you see that your friends maybe dia sangat tinggi, terlampau tinggi, maybe you have this lah. So cek bukan lah. Tapi ini one type of syndrome lah. So normally if the people normally is in primary school lah, normally this kind of man in primary school it already have normally it already have 190 cm punya height already. Sangat tinggi lah primary school your standard 5, standard 6 dia already have the 190 punya height lah. Tinggi dia sudah 190 lah. Sangat tinggi punya. So for this one actually don't too happy lah actually, because maybe it is happen in your body already, maybe you get this disease. Okay, so unusually also, and then the hand is and leg is very long, and then it has the female like breast because it contain XXY. So memang dia akan breast lah, it contain the breast lah, like the female. So actually this kind of people you want to say it is a boy or girl, very difficult lah. So also and then it is sterile lah. Memang sterile kerana dia tak boleh produce the sperm or even ovary. Right. Okay. Another one is female punya triple X. Dia ada 3X. Normal female got double X. Ini ada triple X. Okay, so ini orang ni normally it is the female, memang female because 3X it is totally female. So this one get of the slightly mental retardation and also unusually tall. Ini perempuan ni memang pun terlampau tinggi punya. And that has the wing for learning disability. Yang ni meaning that learning disability meaning that this kind of female is have the not easy to learn something lah compared to normal people. Like kami nak belajar something actually awak ada cikgu awak ada explain macam saya lah. Saya explain 1-2 kali awak sudah faham. Tapi orang ni triple X punya saya explain dia pun tak faham. Sampai saya explain 10 kali 20 kali maybe dia baru faham. Maybe lah. So this is what we call as learning disability. But this one is fertile. Fertile meaning that it can produce the ovum. It can produce the ovum. Yeah. And another characteristic is the the skin is wet and then constriction of aorta, poor breast development. So the breast of this kind of female is does not like the normal female lah. Normally dia memang flat punya lah. Memang flat punya like male lah. Tak macam orang perempuan yang ada breast punya. And then under developed ovaries. So okay, so this one is yang paling penting inilah. Kalau awak tengok yang learning skill dia poor learning skill memang triple X syndrome. And then another one, Jacob syndrome. Okay, so Jacob syndrome is a person who has XYY. 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 Ini memang boy lah. Boy tapi dia tambah satu Y. Memang Y. So actually this one actually it is same as the dia macam normal male. Lelaki ni memang biasa je. Tak biasa usually tu tinggi terlampau. Lepas tu normal sexual development, have hairy ends, have but yang ni lah have criminal tendencies. Kepada orang lelaki kalau mereka buat research lah dekat negara Eropah, Malaysia tak ada lah. Negara Eropah mereka they found that when they take the genetic for those who have the disease, genetic of the pigeon itu orang yang buat penjenayah mereka with the genetic sequence of them, they found that most of them with this XYY punya ada high probability to take a crime, to buat penjenayah. Macam bunuh orang, curi orang, bunuh orang. Memang dia ada orang yang ganas lah. Memang dia ganas punya. Ni memang ganas punya. Sangat suka pukul orang. Memang dia sangat ganas punya. Very, well, we say that is very, not very soft people lah. So orang ni memang kalau boleh sangat suka untuk kawan dengan dia. Kerana dia memang sangat ganas dan juga very easy to angry punya. Kalau dia angry memang dia akan buat perkara-perkara yang kita boleh sangka punya. So ini main characteristic lah. Ini memang main characteristic. And then this one is also delay development of talking, language skill and motor skill. And weak muscle tone. So ini Jacob syndrome. Okay, another one is Turner syndrome. Yang ni pun famous XO. Ini female but it only have one X chromosome only. So this one is that it is ovum is not fully developed lah. So it is sterile. Tak boleh produce the ovum, cannot produce the egg one. So this kind of human female does not have menstrual cycle lah. Tak ada punya lah. Because the ovum cannot form. So normally if you see that this female the face is heart shape face, and then contraction of aorta, and then webbing of neck, and then also both chest with widely spaced nipples, and yang paling famous punya dia sex organ lah, ovum. Memang dia tak boleh produce the ovum egg punya. Okay, so that's all for all the disease. So all of the disease you need to know what are the chromosome involved and also what are the main characteristic involved. So semua kena hafal, memang kena hafal punya. So normally this one will come out in objective or structure. Essay question normally will not come out because essay question sangat kejam lah kalau essay question nak you explain all this. Tapi moon game pun boleh come out tapi kalau memang keluar sangat kejam lah. Boleh cakap macam ni lah Pak. So you need to read lah. Okay. Polyplody. Okay, so so now we already end about aneuploidy. So now we proceed to polyplody. So polyplody actually is the condition when the species originate from an accident during cell division that result extra sets of chromosome. Yang ni memang berbeza dengan aneuploidy. Aneuploidy, it only involve maybe change in one or two only chromosome. Dia memang ada hanya involve only certain number only, very low number lah. But this one is involve extra sets, meaning actually should be 2n. After that, it will become 3n or even 4n. Ataupun it will even become 6n. Because of the mitosis process. Maybe the cell division process, meiosis or mitosis already have the abnormal change already. So it will become 4n, 3n, and 6n. 4n, 3n, and 6n already. So this one is double lah. Dia akan ada extra set. Like human, I give example, human 2n is 46, right? Maybe dia after polyplody, dia akan bagi 3n. So 3n berapa? 3n, you see that 3n will become 69. Or even become 4n. So 4n become berapa? 4n become 92. So this one normally only occur in plant only. Hanya boleh berlaku dalam plant. Animal memang tak akan berlaku. Kalau animal berlaku, memang dia akan jadi orang, jadi monster lah. Itu memang bukan orang lah. Memang dia akan jadi monster lah. Orang yang tak tahu macam mana cakap lah. Memang tak boleh punya. Hanya plant only occur in plant. Animal cannot occur punya. Tak akan berlaku dekat animal. So normally why it is occur? So normally the non-disjunction occur in all chromosome pair. Tadi aneuploidy only occur in certain chromosome. Only one chromosome is occur. But this one all chromosome maksudnya like human 2n equal to 46. 46 semua chromosome ni akan buat semua. Non-disjunction occur semua. Semua occur punya. And then so normally occur in plant. Okay, and then this one also will form sympatric speciation. So what is this? Please go back to chapter berapa? Chapter third chapter, right? Chapter 16. Selection and speciation. So if you know that when you go to the speciation first topic, you will see this term like sympatric speciation. Got two types, right? Sympatric speciation and also allopatric, right? Allopatric and sympatric. Already forget, please go through it. Okay. And then there are two types of polyplody, either is auto or allo. Okay, so auto aneuploidy normally is individual that has more than 2 chromosome sets that are derived from a single species. So normally this is like this. Karyotypic of parent species normally if let's say the 2n is equal to 6, or because of meiosis error, so one of the gamete, both gamete will have the 6 chromosome. Maksudnya dia tak become n lah. Dia still remain 2n. And then after that they go to the they do the fertilization, so it will become 4n. Kerana 2n tambah 2n dia akan berarti 4n. So this one is what we say as auto polyplody. because it is derived from a single species only. Hanya satu species je. Subspecies yang sama. But elo is involved two different species. Maksudnya ibu bapa dia spesies A, bapa dia spesies B. Yang ni ibu bapa dia memang sama saja. Memang sama spesies punya. But for this, ibu bapa dia tak sama. Ibu is A and then bapa is B. Totally different species. And then so if you like that, you see the alloplody punya. You see that if let's say our species A contain 2n equal to 6, and then so meaning that it has 6 chromosomes, and then this one is 4 chromosomes. So after that, they undergo meiosis, normal meiosis. So it will give 3n equal to 3 and n equal to 2. But because and then after that, they go through fertilization. Mereka buat fertilization. So meaning that it will have n equal to 5. Dia bukan 2n. Kenapa? Kerana dia langsung tak sama. Langsung tak sama dengan ibu bapa dia. 2n, kalau ibu dia memang 2n equal to 4, bapa dia 2n equal to 6. Lepas tu, dia anak dia 5. So, kami boleh cakap dia 2n lah. So, we assume that is n equal to 5 lah. And then this one is sterilized organism. Dia tak boleh is sterilized, meaning that it cannot produce tak boleh lahirkan anak lah. Dia tak boleh lahirkan anak punya. Dia mati memang matilah. Tak ada anak-anak punya, anak cucu dia. So our plant actually is very pandai. Memang dia tak boleh, dia pun tahu. Saya sendiri pun tahu. Dia sterilized. So it need to become fertilized. So how it become? It will go through mitotic or meiotic error. It will go through the meiotic error, become 2n equal to 10. Kalau dia original dia n equal to 5, but and this cell plant also very smart, sangat is very smart also. He also know that he cannot survive. Dia pun tak boleh, cannot survive, cannot produce the anak dia. So dia pun tahu. So it will undergo. So dia akan sendiri buat meiotic or mitotic error, buat satu lah mitotic or meiotic error, so that it will doubling its chromosome number become 2n equal to 10. So this one 2n equal to 10, so that this cell is fertile already. Okay, and then this cell is already new species at all, because it does not have the same characteristic between A and B already. Maybe some lah, tapi the chromosome number totally different already. Because A 2n is 6, 2n is 4, but for now 2n is equal to 10, so totally new. So that is why that we call as symplectic speciation at here. So apa tu? Please recap again to your chapter 16. So this is the process of symplectic speciation. So it is also one type of mutation occur in allopolyploid. Okay, so yang paling penting difference dia between these two is one is only involved one species, another one is involved in two species, and then will finally form different species lah. Okay, so that's all for this topic. So finally, our chapter 17, almost 1 month, already finished, totally finished. Okay, so next chapter I will proceed is chapter 18 lah, gene technology. So I don't know your school teacher already teach until that chapter or not. I don't know, because I don't know. So okay, so any question? No question. Wah, semua cemat tau. Boleh score lah. Ini kalau MPM keluar soalan chapter 17, full marks lah. Kan? Memang akan makan penuh kan? Kerana semua cemat. Tak ada question, tak ada soalan. Okay, so no questions. So we will end the class now. So we will see you next Friday. So thanks all. So have a best and good night all. Bye.